Labiotech Hangout. I'm here this month with the founder and CSO of Elypta. Hi, Francesco.
Hi.
So Elypta is a diagnostic startup based in Stockholm. You're developing a liquid biopsy platform based on studying cancer metabolism. So you were started back in 2015 when you were doing your PhD and your postdoc, and then you incorporated in May 2017, raised the first round of capital, and now developing the technology as well as the company. But I looked at your story. It's really interesting, and it's pretty rare in Europe to have someone finishing his PhD or postdoc and then starting a company. Can you walk me through the story and how you got started?
Sure. So I joined Jens Nielsen lab at the Chalmers University of Technology, that's in Gothenburg, so the other side of Sweden, and as a PhD student. And Jens is directing probably one of the largest systems biology group in Europe. And at the time, I was coming from a chemical engineering background, and Jens was mostly interested in switching from studying a very simple organism like yeast to studying metabolic diseases in humans, essentially using the same tools. And he put me on a project in which we were supposed to look at cancer metabolism using a systems biology perspective. And it was very early on in my PhD that I was, applying essentially this data analysis tool. And we recovered that there was something particularly interesting in one form of cancer, which is called renal cell carcinoma. It's the most common form of kidney cancer. And this was always a side project for me. I mean, I started working on it pretty much as, a potential study that could be translational in one day. And it really took off when I started collaborating with a hospital in Italy. This clinician, I would say, was a bit of an evangelist. He took at heart our approach, our systems biology approach, and started collecting samples for us. And really, along the years, this was never my main project. We realized that we were on top of something because we got the data back, and the samples we were collecting were, blood and urine. And we realized that the particular class of biomarkers that we recovered from our analysis of metabolism was extremely powerful. It could distinguish healthy people from kidney cancer patients, essentially, with 100% accuracy when you combine the blood and the urine. So it was at the end of my PhD, it was in 2015, that I was looking for a job in management consultants. And then Jens called me up in his office, and then he said, you have some really good results there. Why don't you go down the road of innovation? To be quite fair, I really didn't know what I meant by that. I never really been so interested in startups. I think I was entrepreneurial in my way of doing science, but not really as a business. And that's when things essentially started. I mean, it was a long journey. And essentially 2 years later, actually, this startup project became an actual company. Was, as you said, in May 2017.
Yeah, no, that's great. And can you give a bit more details on how was that transition in a daily job to transition from in your lab work to leading the whole project? How was that?
Yeah, this is interesting because this is a diagnostic company, so I mean it's incredibly science intensive. It's based on a new technology, a new discovery. So the amount of work that you need to do from the business side should be incredibly hard and very, very intense. But in the beginning, I think my biggest challenge was really in the beginning to transition from a science perspective to a business perspective. So in the beginning, I was actually doing the same as before because I was thinking that the more we nail the science, the better chance we have to build a business. And so in the beginning, I really didn't notice any different. I was almost complaining that there is nothing different. It was, I think we participated to a competition which is called One Start, a great competition, by the way. And that was when we got this 1 or 2 day mini MBA because we were chosen as one of the semi-finalists at the time. And that's when I kind of realized that, you know, focusing on the science was not the only thing. I started working more on essentially talking to people. That's the biggest difference. You start going outside your desk and asking for advice, how you should do this and who is going to buy it. And you talk to your prospective clients. It's that's the biggest change. Like you go outside.
So you really learned on the go learn by doing. You didn't take any special program or entrepreneurship program.
No. It was I winged it most of the times, made tons of mistakes on the way. Fortunately, nothing critical. But I mean, yes, I could say that. And I mean, I can't. You know, I can't underscore as much as now how important it is to talk to people. I mean, they are the ones who saved this venture in the end, because my knowledge was obviously not sufficient.
I mean, I completely agree with you on the mistakes we do with them every day. And what I used to say to my team is, it's fine to do mistakes, just don't do them twice.
Yeah, I agree.
That's cool. And so, One Start, was that in the US or in Sweden, the competition?
No, One Start had this Europe and rest of the world. It was essentially everything outside North America.
Okay, because it's originally an American competition.
Yeah, I mean, this was in London. It was in 2016. I think now it's discontinued. It was an excellent thing. I don't know what they stopped it.
Okay, that's really cool. And so now you are 2 years into it and working full time. How has it been?
Fun? It's been fun. It's been tough. I mean, I confess I was not super hard working during my PhD. I think I was very lucky in terms of some experiments that panned out at the first try. But transitioning to business, then you realize that that there are a lot of things that actually depend on you, and they, you know, when you're doing research, you're doing research. That's pretty much all your activities are directly into research. Okay, you can have some teaching assignments, some conferences to go. But all of a sudden, when you transition to business, you have to take care about intellectual property rights, financing. You have to do business development. You have to do, you know, simply market research, market survey. You have to manage people. I think that's by far the most complex part for a PhD student. Europe essentially used to just deal with yourself and your own projects, and you deal with your collaborators if you have any. But all of a sudden, you have people reporting to you, and you're supposed to give them direction and vision. That takes a lot of time, but it's by far the most satisfying part, because you're dealing with humans that are connected by the same mission as yours. It's a fantastic feeling.
That's good. I can definitely, I definitely agree with that. That's good. And so to get back to the lab where you were, so Jens Nielsen lab in Gothenburg at Chalmers. It's one of the very few labs I know in Europe where entrepreneurship is encouraged. I mean, there's a few others, but it's really encouraged, and I think your example is really speak to itself. And when I was in Boston, was really striking to me. All the big labs, like I visited I interviewed George Church, and same, he was saying, okay, half of my students are founding startups. That's amazing, and it was very striking for me when you compare it to European labs where it's academia, and do a career in academia. Can you go a bit deeper in the like the culture inside the lab and how it helped you to get started?
Yeah, sure. Well, you're hitting on one of the main differences between Europe and America. I mean, like it's the willingness to translate findings into business cases. It's enormous in North America. I think Europe is definitely lagging behind, but I think it's catching up. And to one point about Sweden, like Sweden has this brilliant law about the teacher's exemption that gives researcher ownership of their own intellectual property, which means that you don't have to go through tech transfer offices to license your own discovery from the university. You own it as a researcher. It comes with some drawbacks, of course, but I mean, that I think will give Sweden an edge. And I think Jens has really made the best out of it. He, I think, spun out 4 companies, which, you know, in biotech in Europe is I would say I agree with you. It's a large amount. I'm not sure if this is uncommon or not. I agree that the focus seems to be a lot about academic work in Europe, but I don't know how many labs now are changing, switching gears. So that I cannot comment on. In terms of culture, I would say that still the lab is still very much focused on science, and of course, like it's in a technology university as Chalmers, so obviously there is always an eye towards applications. I think that helps a lot. And Jens, of course, he has the experience. I think once you do one biotech company, you're very much prone to repeat yourself. I mean, so.
That's cool. I like the comment on the IP. It's something we look quite a lot on the CRISPR case, and I received an email this week on saying that I looked at the list of the owners of the patents. Actually, Charpentier is as her name because she was doing the work at Umea, and
right.
so she owns patents herself on CRISPR, and then founded her own holding company to license it out. It's pretty interesting. So I guess this helped as well on like spinning out the company, and then did it help also to attract investors afterwards?
Well, I mean, I would say I can only speak for Chalmers because Chalmers has an interesting status in Sweden. I mean, it's not entirely public. There is a foundation that owns, so it doesn't. It probably has its own advantages to be in that kind of university. So what happens there is that Chalmers, I think, pushed very strongly on entrepreneurship. They have one of the, as far as I know, one of the best incubators in Europe. I might even dare to say the first, and one of the best accelerators. They have Chalmers Foundation, I think, maybe I'm getting the details wrong, but gave a fund of 300 million crowns, around 30 million euros, to the how do you call it, the umbrella company, Chalmers Ventures, to
invest in
fuel
from
the university. And we're an example of that. Chalmers Ventures is actually our first investor. We got our pre-seed money, thanks to which we could file the initial patents. As you know, in life science, this is everything, right? So, and again, this actually speaks for the teacher exemption. It's great because I own my own IP, but at the same time, I have not so much money. So paying for attorneys and stuff, I can't really do it out of pocket. So you kind of fall back in the university system, but it's very well oiled. They give you money to support the filings, and of course they take a stake. So they're the first investors actually. But I'm happy that the system exists. I would have never done it myself if I were on my own.
Yeah. Makes sense. I mean, it's always you need public help, and when it's well done, it's great to get you started. But yeah, don't want to be too dependent either. So it's good.
Don't want to spend 6 months negotiating with the tech transfer office. I mean, that seems very unhealthy. And I know it.
6 months. 6 months is fast.
Oh yeah.
I know from talking to other founders in life sciences, it can take ages and very unfamiliar terms, and you talk with people who don't understand really how you will like evaluate your patent. It's pretty nonsense to me sometimes when I hear the stories, but it's good that it's working differently in Chalmers, and you have some university doing it correctly. Sounds good. So let's just switch a bit to Elypta and the technology. So from my understanding, you base analysis on cancer metabolism or metabolites and a broader analysis versus just a single biomarker. Can you expand a bit on what makes it unique and how it works?
Sure. So, I mean, our technology belongs to a class of technology that you would refer to as liquid biopsy. So essentially the ability to make diagnosis or certain decision that essentially carry diagnostic information using blood or other fluids that primarily involving cancer patients. And so this is essentially the if you want, the industry segment we belong to. And it's brilliant because, like, right now in cancer, essentially, diagnostics is pretty much done, I'm simplifying a lot, with medical imaging, computed tomography, MRI, ultrasounds, and then
and solid biopsies.
And solid biopsy, when you actually know that there is a tumor and you know where to put your needle, with the exception of the blood cancers, of course. But I mean, the key aspects here was that we were essentially looking at the last layer of biological information in a cell, which is metabolism. While most, if not all, liquid biopsies currently under development, they look at the first one, DNA or RNA, genetics. What's the difference? Well, I think the two approaches are essentially orthogonal. When you find DNA in the blood and there is a mutation, it's very unlikely to be any other disease than cancer. There are very few other diseases that actually shed DNA in the blood and that carries the information about how the disease started. So these are extremely specific diagnostics. I mean, if you find it, it's very likely to be cancer. On the other hand, you need to have a quite big tumor before the DNA is shed into the blood.
Okay, so it's mostly for stage 2, stage 3, and you cannot detect very early tumors.
For example. While when you look at metabolism, you really look at what has happened in a cell. DNA will tell you what will happen. And some of these processes are essentially triggered in the beginning. If a tumor cannot rewire its metabolism, it cannot find oxygen, it cannot find glucose, it cannot find a way to essentially remodel its microenvironment to survive. The tumor will die out, right? So these are processes that are very contingent to the evolution of the tumor. And we are exploring one of those processes. And there are, of course, metabolism is huge. There are, we always taught in school that there is a glycolysis and, probably 20, 30 compounds. But in reality, there are over 3,000 metabolites that make up the network. And we look at the one that is actually a panel. We don't look at one biomarker. We look at a panel of biomarkers that speak for a specific process that is important for cancer metabolism. That happens to be essentially can be measured in the blood and in the urine. And that's what we pick up. So our sensitivity, as opposed to the specificity, is exceptional. We just published a paper in kidney cancer. We can see stage 1 to stage 4 renal cell carcinoma with, I think, 93.5% sensitivity as opposed to healthy controls.
Okay, what's the sensitivity of your competing tests?
That's interesting. I honestly, I think these are all under development, so the figures are probably not released. I know there are some papers that explore kidney cancer, so renal cell carcinoma. I would guess it will probably be around 50%.
Okay, because that was one of my questions as well. I mean, I know a few diagnostic companies, and one of them actually doing epigenomics marker, and actually the sensitivity is pretty high. I mean, in blood and epigenetics marker, but I was wondering how much more sensitivity you can bring. The 93%, if you can do it on a large quantity of samples, it's pretty high actually. I think the main challenge of liquid biopsy or diagnostic in general is I mean, the test and the science is one part, then how you access the market is another part. So what's your plan on that side?
Well,
what's your plan?
It's a great question. Sometimes I ask myself, why did I end up in diagnostics? Like, it's almost as expensive development costs as okay, I'm exaggerating, as therapeutics. I mean, it's very expensive to develop a diagnostic technology, let's put it like that. But the reward compared to therapeutics, like, if you come up with new approved drug, it's much smaller because if you want, it speaks from our mentality. It's easier to treat rather than prevent, and you're willing to pay more. So, of course, like, in the end, it's all a matter of health economics. Like, if we target specific applications, we don't. When I speak about a blood test for cancer, many people think about, oh, great, I can go to the lab, give my sample, and see if I have cancer or not, and I'm perfectly healthy. This is a very tough scientific question and our test, as much as genetics-based liquid biopsy, were very unlikely to be able to do this one day. I don't know yet. The data is not there. But what we can do is to help patients that already have cancer, and this is actually an emerging problem. Like the patients right now, cancer is becoming a chronic disease. Like we now live from first diagnosis around 5.5 years, which means that we need to be followed up as cancer patients like for 5 years. It's a long time. And if we can replace or complement what we have now, which is imaging, with anything that can follow up, if I go surgery, will the cancer come back? If I'm taking drugs, am I responding to the drugs? Can I simplify this process? And since our therapies are becoming better and better, these 5.5 years will become longer and longer. So I guess at a certain point, it's a matter of market need. It will be so high that diagnostics will necessarily become a business, a very viable business.
Hope so for you. I think it's a challenging part, which is pretty. I mean, it's pretty crazy on the diagnostic. We had a panel yesterday at our meetup. They're doing a diagnostic test for multi-resistant bacteria. They spent 100 million to develop it. It's pretty a big amount. But now there's the access to market, FDA approved in Europe as well, and commercializing with hospitals. And it's one of the few diagnostic companies in Europe who are very successful at curates in Germany. So I hope, will be the case for you as well. But I think it's a few years down the road as well. And what's in the space in liquid biopsy, you have quite a few competitors, actually, or companies going in the space. I mean, not to mention the very big ones like Grail, but a few others seems pretty hype right now. So what do you see as your main competitors, and how do you compete with them?
I mean, on a general level, the liquid biopsy space is becoming very crowded, and of course, like the big players, like the one that you mentioned, are certainly competitors. When you look at the potential of our platform that has been tested primarily in kidney cancer, but we have data in a variety of indications, and they're equally promising. So we're sitting on something particularly hot. But when I look at them, the main issue will always be, what are you measuring, right? I mean, as long as you're measuring that layer of information, genetics, you will get this kind of information. I mean, you can't extrapolate too much from it. We're orthogonal. I mean, we're looking at process that, a process that is in metabolism that speaks for cancer in a different way than genetics does. And I think that's gonna be the main differentiator. Our possibility to, so far seems like that the biggest advantage is in its exceptional sensitivity to essentially every type of cancer that we tested. There's one thing to note here. Our lead indication is kidney cancer, which is, I think, a bit unusual. It's not one of the big, prostate, lung, colon, breast. And the reason being that, well, first of all, the data, the science was strongest in that indication. But also, when you look at that, kidney cancer is not really a small market, and there is nothing blood-based available right now in the routine. So we are tapping into an area that has an incredibly high clinical need that is unmet, because, the big company are mostly interested in the bigger cancer types. So the research has been going on in kidney cancer is relatively limited, which is a pity, because, I mean, 180,000 patients every year in the US, in Europe. It's not a small population. And they all want to know if the cancer comes back after surgery. They all want to know if they're responding to drugs. We ought to do something, right?
No, makes sense. I mean, it makes sense to start with a small indication and then prove the technology on that, and then expand to the other cancer. So, and as a last question, we have to wrap up, running out of time. But what's the next steps for you for the company?
Well, I mean, I guess our biggest challenge is really to prove ourselves, because like going into metabolism is essentially going against the flow. I mean, I'm not aware of any other liquid biopsy technology that is tried and successfully proved with data that this may work. And of course, we need to validate what we have. We need to develop the liquid biopsy platform based on metabolism that we can be certain will help patients. When we have that, and it's, if you want, both a technical and a clinical challenge, but when we have that, then we are ready for raising our big Series A round and proceed to the clinical trials that are, you know, the one that will give us FDA approval. So that's the plan. Knock on wood. I have wood. So, yeah.
Look on wood as well. That's great. Great. Thanks. You have to catch up a train, so I will let you get that.
Yes.
Thanks a lot for joining. Really interesting story. Great startup, and good luck with it.
Thank you so much. Thank you so much for the invitation.
Thanks. Bye bye.
Ciao.